So good afternoon, everyone, and welcome to the Palaeoverse Lecture Series. This lecture series is focused on software development and methodological approaches in paleontology with the aim of promoting the use of new tools in the field and to give researchers a platform to showcase their work. So with this series, we hope to shake up the usual type of lecture by having a mixed format with time dedicated to presentation, demonstration, and discussion of software or methods being presented. So typically these sessions are going to run for about 50 minutes in total, and we're running this series throughout the year on the last Thursday of every month. This is the final lecture in our summer series, and we're really excited to announce the new lineup of speakers, which is shown here on the screen. And note the time change. They'll be held at 1600 UTC, with the exception of the talk on the 21st. And we're really trying to accommodate as many time zones as possible with this time shift, so bear with us. The lineup will also be announced on social media soon, so keep an eye out for that. And we put the registration link below if you'd like to sign up for the next block of talks. If you're interested in presenting a new method or software that you've been developing, please feel free to reach out and we can include you in future versions of the lecture series. So before moving on, just a few quick introductions. My name is Erin Dillon, and with me today I have Lewis Jones, Bethany Allen, and Will Gertie. The four of us are on the development team of the Paleoverse, an organization aiming to bring together the paleobiology community to share resources, reach agreed standards, and improve reproducibility and paleobiological research. So if you want to know more about the Paleoverse, please check out our GitHub organization page and website, which I believe are being put into the chat right now. So that's all from me now. Bethany will now give you some helpful tips and instructions for this webinar, followed by Lewis, who will introduce our speaker this month. Thank you very much, and please enjoy the lecture. Over to you, Bethany.
Thank you, Erin. So as Erin mentioned, the format of this seminar will consist of a talk by our speaker, followed by 10 to 15 minutes at the end, dedicated to questions and answers and discussion. My fellow hosts and I will help field your questions and moderate this discussion. Please note that this seminar will be recorded and will be made available on the Palaeoverse YouTube channel. However, we'll stop the recording before the Q&A period in case you prefer not to be recorded. Before we get started, I'd like to quickly point out a couple of useful Zoom controls at the bottom of your screen. So there's a Q&A button. You can use this to submit questions to our speaker. During the Q&A session, we'll either read out your question or invite you to turn on your microphone if you'd like to ask the speaker directly. There's a raise hand button. Alternatively, you can raise your hand and we'll call on you to invite you to turn on your microphone so you can ask your question. There's also chat. Currently, you can only chat with the host in case of technical difficulties, but we'll allow you to chat with other attendees during the Q&A period. And also closed captions, which you can turn on and view the transcript at any time. Lastly, I'd like to highlight a couple of ground rules. Our aim is to cultivate an open, respectful, and inclusive environment during this lecture series where we can learn from each other. With this in mind, we ask that you please be respectful and kind when engaging with the speaker and fellow attendees, particularly when asking questions and interacting with others in the chat. Any form of harassment or inappropriate behavior will be grounds for removal from the webinar platform. For more information, please refer to the Palaeoverse Code of Conduct, which can be found on our website, and I'll also put a link to that in the chat. And with that, I'll pass over to Lewis to introduce today's speaker.
Thanks, Erin and Bethany, for getting us all up to speed on how the lecture series will work today. So today I have the great honour of introducing Dr. Euan Furness. Now, Euan completed his PhD at Imperial College London, where he worked extensively on using individual-based mechanistic computational models to disentangle the drivers of spatial gradients of biodiversity. Euan is currently a postdoctoral researcher within the Deep Time Ecology Research Group at the University of Cambridge, and his current research focuses on developing integrated models that capture the myriad of interactions and dynamics associated with the Ediacaran to Cambrian transition. Today, we're very excited to hear from Dr. Euan Furness with his lecture on reproducing emergent eco-evolutionary patterns from first principles agent-based model. Please join me in welcoming Dr. Furness. Over to you, Euan.
Well, thank you, Lewis, and thank you to the other organisers as well for inviting me to come and give this talk. I hope you can all hear me and see everything okay. I'll assume as much. I want to talk to you today a bit about this piece of software that I used extensively during my PhD period. It's a eco-evolutionary simulation tool that functions at the individual level that has great potential to be used to disentangle drivers of diversity and the mechanisms underpinning the diversification process, as well as to answer other eco-evolutionary questions. So today I want to talk you through a bit about what RevoSim is and a bit about what it can be used for, and then actually give you a demonstration of quite how straightforward it is to ask questions in RevoSim and how you might be able to use this in the context of some of your own research. So anyone who's seen me give talks before might well have heard me talk about RevoSim, but I'll cover first just what RevoSim is and what the rationale is for why it exists. So we are interested in studying the evolutionary process. We are interested in studying the dynamics and the results of evolutionary processes. And there are a variety of ways in which we can do that. So one of them is to study evolution at the relatively short timescales. So these are processes that we can observe in the modern. We're talking timescales up to the decades, maybe up to 100 years, where we can see populations change in terms of their genetic composition or in terms of their phenotypes, and we can perform experimental manipulations upon those populations, and in so doing, we can learn about how evolution functions at those short timescales. At the same time, we have methods for studying evolution on very long timescales. So by collecting fossils from geological units, we can study how different groups of organisms have evolved on the timescales of many millions of years, and we can infer from that the evolutionary processes involved. But there's a disconnect between these timescales, and that is these intermediate timescales. So we're talking things of timescales of hundreds to maybe low millions of years, where the fossil record often won't provide us with the level of resolution that we need to answer questions that we might be interested in about processes that are occurring in this window. But we also can't perform experimental manipulations or observations over these timescales because, well, we can't be around long enough to do it. It's just not possible to set up those experiments. And so when RevoSim was being developed, it was being developed as a tool for filling in these intermediate timescales. Because in essence, what we can do with RevoSim is we can, or any other kind of individual-based eco-evolutionary modeling tool, not necessarily just RevoSim, but that's the one I'm going to talk about, because we can speed up the clock effectively that we're looking at, we can impart all of these relatively well understood ecological and evolutionary processes from short timescales onto systems that we can then run for longer timescales and see what emerges from those systems as a result. So that's basically the rationale for why we have this thing and the setting in which it is useful. Now, if you want to do simulation of ecology and evolution, you've got a wide range of options for tools you can use to do that. And they come in a variety of flavors. So starting with the sort of coarsest of them, you have these things like diversity-based models, where you're assuming that every species within your system is fungible. All your species are identical from a functional perspective. And while you might care about the number of species in the system, you don't need to care about what each species is doing relative to the others. That's a very simple thing to model. And because it's so simple, you can model it in the context of much more complex processes in other elements of your model. So for example, this Semeno et al. 2022 paper, they used this diversity-based approach where they modeled the abundance of species through time in different locations, and they coupled that with complex reconstructions of the climate and of sea level and of continental configuration and so on, and used that to estimate diversity through time. If you want to take one step finer, you might want to look at population-based models. So instead of assuming that all of your species are functionally identical, you might assume that actually the species have relevant features that are different from one another. Perhaps some species are more capable of surviving in one environment than another, for example. And if you want to model that, you can do that using population-based models, where you say, well, all of our individuals within our species are the same. But the populations of different species, they can be different in relevant ways. And so you could have a spatially explicit model, for example, where you have grid cells that each contain a certain number of a certain species, and that species has certain properties. And the Genesis model is an excellent example of this, and it's been used to do things like study change in diversity through time over periods of tens of millions of years in South America, for example. But if you want to take one step even finer, you can turn to individual-based models. And where the population-based model had to assume that all of the individuals were functionally identical, or at the very least, that any variation within those individuals could be captured by some set of summary statistics, your individual-based model will track every individual separately. So there's no requirement for them to be the same or to have any easily predictable distribution of variation. And RevoSim is an example of this individual-based approach. So the original paper describing this system was Garwood et al. 2019, and we published another paper, a follow-up to that with some new features in 2023. And all of these different approaches have their costs and benefits. Broadly, as you get more fine-grained, as you go from diversity to population and to individual-based modeling, you are increasing the amount of computational effort required. But once you start modeling things at an individual-based level, there are significant upsides as well. So because every individual has its own genome, for example, our speciation mechanisms are not imparted upon the model, but rather emerge from the model organically. So for example, instead of having to say two populations will become different species once they've been separated from each other geographically for a certain amount of time, we can just say, well, whatever happens to the genetics happens according to the rules that we believe to be true and which are incorporated into the model. And actually, when a species arises is a property of how quickly the genetics diverge between those populations. So it's an emergent property rather than a hard-coded rule. There are also things like we can study different speciation modes. Instead of having to say, well, all of our speciation will occur by allopatry, it's perfectly possible for things like sympatry to occur in RevoSim because, again, we are not imparting a rule upon the system for when speciation occurs. We're just saying, well, once you have sufficient genetic distance between two groups of organisms, you can consider them to be different species. And because we're modeling all of the individuals separately, the individual variation within the population can organically influence the evolutionary trajectory of its populations. And that's something you can't capture unless you are actually tracking the individual variation. So how does RevoSim actually work? Well, I want to cover some of the basic rundown here, and then later on I'll show you a kind of example of what we can do with it. But it tracks different individuals as different entities within the model, and it can track up to 1 million of them at a time. And it can run these simulations at a rate of multiple generations per second. So that's a very large number of organisms turning over within the system. All of this is possible on a standard desktop or laptop computer. There's no high-powered computing necessary for this. And that is basically down to Russell Garwood, Alan Spencer, and Mark Sutton, who were the original designers of this thing and created some very nifty tricks for making it all run very quickly. So credit to them for that. The individuals within RevoSim are composed of genomes, which are some multiple of 32 bits long, and they are binary. So instead of a ternary genome with different DNA bases, we just use zeros and ones, but from a fundamental perspective, that's more or less the same thing. We just have variation within the genome representing changes in base pairs effectively. And you can think of these 32-bit chunks as equivalent to chromosomes if you want to. Every individual in the genome lives within a cell in a square grid, so this is a spatially explicit system. And every one of these cells has 3 environmental variables, which we can define using the red, green, and blue color channels of an image. So you can think of these variables as equivalent to altitude or temperature or rainfall, but really they're just abstract variables. They don't have any imposed meaning. Suffice to say that 2 cells that have very similar environment colors, functionally speaking, have very similar environments. And all of the organisms in the system have a fitness, which is this term defined by an interaction between their genome and their environment. And what this fitness does is it dictates the rate at which organisms gain energy from their abiotic environment. So any small change in the genome of an organism will result in a small change in its fitness, Any small change in the environment color will result in a small change in the fitness of any given genome. And there are many more ways to be fit in any many more ways to be unfit in any given environment than there are to be fit. And all that's relatively realistic. So what this looks like when you try to analogize it to the real world, one of the best examples, and this is one that Mark Sutton likes to turn to, if anyone who knows him will know that he loves his brachiopods, are these brachiopod type ecosystems. So the organisms are sessile. They are gathering energy from their environment by being adapted to their environment, and their interactions with the organisms around them is limited to competition for the resources that are funneling into that environment. Although in the more recent versions of RevoSim, we've actually added more complex interactions as well as an option. So these organisms are gathering energy from their environment at a rate dependent upon how well adapted their genomes are for that environment. They use that energy to breed and produce genetically similar offspring. This breeding can be asexual, or it can be sexual, or it can asexual or facultatively sexual, sorry, or obligately sexual. That's all settable within the simulation. And the organisms, if they're breeding sexually, will produce offspring that are a combination of the parents' genomes. And then mutations will also occur during breeding, which will result in the offspring being slightly different. And because we have mutations, we have selection based on the difference in fitness between organisms in environments, which is based on their genome. We will naturally get evolution, and organisms will tend to evolve towards being fit in the environment in which they're found, which is what you would hope for from an eco-evolutionary type simulator. You can also change the environments through time, and if you do that, the organisms, the populations of organisms will evolve to track those environments and remain fit in the environment in which they find themselves. And because of the sheer number of genomes that are possible, there are a large number of genomes that are optimally fit within any given environment, that are gathering energy from the environment at the highest possible rate that they could. But there are many, many more genomes that are very unfit. And this does make sense. If you think of a shallow rock pool, for example, it would be perfectly possible to be fit in that environment by being an alga, or by being some species of snail, or by being a crustacean. But there are many, many more organisms that would not be very happy if you submerged them in a shallow rock pool. So it's a perfectly realistic thing to have, and it's something that was in the mind of the people who were putting this together. The actual number of genomes you can have is 2 to the power n, where n is the number of genes, which is some multiple of 32. So often 64 or 128, and that suffice to say that's a very large number of possible genomes. This is a little piece of footage I like to show of RevoSim in action. So on the left here we have the environment. You can use any 100 by 100 image as the environment, and you can change the image through time. So functionally speaking, you can use a video as an environment if you want to, and that's what we have here. Every pixel of this image is a cell, and the difference in color between the pixels represents different environments. And on the right, what you're seeing is a color representation of the genomes of the organisms in the environment. So similar colors on the image in the right represent similar genomes. And you can see that organisms will tend to expand their range to fill similar areas of color. And then over time, if the environment remains relatively stable, they will specialize to become adapted to the environment that they're in. But if the scene changes rapidly, in other words, if the environment changes rapidly, then you will see extinctions. So this is all suggesting that the organisms are behaving in a sensible way ecologically. They're tracking their environment, and they're evolving to become fit in their environment. So that's mechanically how RevoSim works. What does that mean in practice? What can we actually use it to study? Well, all sorts of things. And I hope to suggest to some of you that this might be something you want to use to look at some of your own research questions. And if anyone is interested in that, I would encourage you to get in contact with me or in contact with Mark Sutton or Russell Garwood to talk about the possibility of doing that. And there's even the option to incorporate more stuff into RevoSim as necessary, to add features that might be necessary to study more things. This is still a piece of software that is undergoing change. So one thing we've used this to look at in the past is drivers of diversity. So I did my PhD, as Lewis introduced, on a lot of diversity gradients, and most specifically the latitudinal diversity gradient. And one thing I did was I used RevoSim to look at specific drivers that had been put forward as potential drivers of the latitudinal biodiversity gradient, such as productivity and environmental stability. And because we have a model system, we can artificially manipulate those features within the system and see how that affects the diversity of species that we recover. But we can also look at diversity on a much smaller scale. So here we have some island biogeography, and because what island biogeography requires effectively is dispersal of organisms through space and adaptation to their environment, and then extinction, that's all features that will emerge from our RevoSim simulations. And so this is another thing that we can look at and something we have looked at. Something that has been looked at a little bit, but I'm actually very interested to look at more in the future, is this idea of the speciation modes themselves. So within RevoSim, we can explicitly observe one species splitting into two, for example, which is not something that's particularly easy to do in the field or in the fossil record. And because we can observe that, we can observe how that species is changing through time as well. And so we can look at things like punctuated equilibrium. And that's, yeah, something that we're still working on, actually. And finally, or not finally, one other possibility is predator-prey ecological interactions. This wasn't something that was included in the original publication of RevoSim. It's something we have now added, the ability for organisms to not just gather energy from their environment, but also from one another. And we see when we incorporate this into RevoSim that lots of organisms will gather energy from their environment, but several species will start to emerge, especially when productivity is high enough, which will be obligate predators. They are completely incapable of gathering energy from the environment, but they grab it from the organisms around them, and they are optimized to do that. And we also have the capacity to look at things like pathogens. So this isn't something I know too much about in RevoSim. It is a feature that exists. This is something that Russell Garwood has been working with. So if you're particularly interested in that, I would encourage you to talk to him about it. So that's sort of rundown of what RevoSim is and what we can use it for. I'm going to show you now what the process is of setting up a simulation in RevoSim. It's an incredibly easy piece of software to use. It has a lovely user interface, which makes it quite straightforward. It's also very easy to download, and if any of you want to, you can actually go to this link here, the Palaeoverse RevoSim GitHub, and go to releases and download RevoSim right now and follow along with what I'm going to be talking about, because it really is very straightforward and it's quite a powerful tool. It comes with EnviroGen, which is a separate but related piece of software, which effectively exists to generate 100 by 100 images to use as environments in RevoSim. So what I'm going to do is crack open the software now and show it off. Everything that I'm going to be showing off now is just what I have downloaded from that link. So there's no other scripts, there's no other pieces of software involved. It's all running out of the box from that download. So I encourage anyone who's interested to go to that now. So if I'm just briefly going to stop screen sharing so I can set this up. Where's my mouse gone? Ah, there it is. Okay. Okay, there we go. So if I go to this and put this over here, then I can share the bigger screen. Okay, I should be sharing again now. It's here. So all I need to do is open up RevoSim or EnviroGen. I'm going to start with EnviroGen. I'm just going to show you how to generate an environment if you want to produce something that you can then feed into RevoSim and get it to run a simulation. So this is the EnviroGen program. It's relatively straightforward. There's a file path on the right here where you can control where you're going to save your files as they're generated. You can choose how many images you want to generate. You can choose how big they are, but 100 by 100 is probably where you want to be. And then there's all of these various different environment types that you can generate. I'm not going to go through full details of exactly how they all work, but noise is fairly intuitive. It's going to generate a 100 by 100 image where every pixel is independent in color from the pixels around it. That's going to be a very messy environment. There's no spatial structure to it. So if you're trying to do kind of ecological considerations about habitat size and that kind of thing, that's maybe you want to. There's some reasons to do that. For example, you could have if you have another environment, you could overlay the flat color on top of it, and then you could increase that through time, and you would effectively be increasing one of your axes of environmental variation continuously through time, like temperature, for example. There's a tool here to combine two stacks of images. So as I just suggested, you might want to increase something through time. Well, you can do that here if you want to. And then there's these two tools here, which are different environments that you can generate that all have some amount of spatial and temporal structure to them. So I'm going to generate one of them now, and you'll be able to see what that looks like. So essentially, these are composed of blobs of color that will overlap and will move through space and interact and produce a spatially structured environment. So these settings control some of the features of the environment. So the number is just the number of different colored blobs. We're going to make that quite high. We'll make them size velocities, how quickly they're expanding or shrinking in the image. So we'll make that a bit lower. Is there anything else you want to change? No, I think that's fine. Okay, so we'll just run that. And all it's going to do is generate a stack of 500 of these images, which we can then feed into RevoSim. What's that? Oh, there's a little progress bar at the bottom, but it's hiding behind me at the monitor. There we go, that's done. So I'm just going to close EnviroGen down now. It's done its job. But if I wanted to, I could generate a stack of noise, for example, and I could combine that with the light stack I've just generated, or I could create a stack of a flat color and combine that. So EnviroGen's a versatile little thing. And now I'm going to open up RevoSim itself and guide you through sort of how that works. So this is the GUI for RevoSim. It's got a bit more to it than the EnviroGen GUI. It's got all of these different options at the top here, and then it's got this stack of things here that are worth commenting on. So what we have here on the right is our environment image. So that stack of lights images is what we're going to use here. But these three blue vertical stripes is the default environment for RevoSim. You probably don't want to use this for any practical experimentation, but it has to load in with something, so this is what you get. And on the left here, what we're visualizing is the organisms in our system. So at the moment, most of the screen is black because most of the screen has no organisms in it. There's only the very central pixel here, which has 100 organisms, which are the organisms that have been seeded into the environment at the very start. At the bottom here, we have a series of summary statistics, which tell us a bit about what's going on in our environment. So at the moment, it says 0 organisms. That's because it hasn't started yet. There are actually 100 of them in there. Up here, we have various tabs that I'll lead you through. So first of all, logging. If we're going to use this for experimentation, we need to make sure that we're generating some output that we can use. I mean, it produces lots of very pretty images, yes, and I don't mind showing those off, but from a scientific perspective, we want something a bit more quantitative. So what we can do is we can go to our logging system here. We have a file path, which is where it's going to send the logs to. We have the refresh rate, which is how often it is going to generate a new entry in those logs. So 50 means every 50 iterations, which on the default settings is about every 2.5 generations in the simulation. And then we have all of these options, which control whether or not we are logging particular images from the environment. Usually we don't need these, but if, for example, we wanted to have a visual record of the distribution of all of the species within our environment through time, well, we could tick the species box and we would get that. I'll leave this ticked for now, and I can show you later what that looks like. If we go down here to the running log section, we can tick this little box, which will make sure that we actually are producing a text log in a file, which is we're going to need to. And then there's a lot of things you can put into here. These 3 boxes control exactly what is put into our text log. The header text is printed at the start. The iteration text is printed every time we have a new logging iteration, so that's every 50 iterations. And the species text is printed for every species in those logging iterations. In order to control exactly what is produced, you need to know all the various keywords that you can enter into these boxes. But you can pull those keywords off the documentation for RevoSim, or you can just press v2 log, and there's a sort of default set of keyword options that it will give you, which are useful for plenty of things on their own. We'll run it with this now, and you can see what that looks like. If we go to settings, we can modify things to do with the organisms and the environment. So if I go to the organisms tab of the settings bar, we can control the mutation rate of the organisms. We can control how long they live for in the simulation. So higher values of this will be effectively organism lifetime. The breed threshold and breed cost is how much energy the organisms need in order to breed. And then the max difference to breed is the maximum genetic distance between organisms in our simulation that they will be able to breed over. So if we have this very high, then we're not going to get the generation of lots of species. If we have this low, then we will get lots of species generated that just from very small genetic changes. So 3 is a sort of compromise on that. And if you hover over these boxes, you get a little tooltip that gives you some information about what these settings mean. We can control the mode of breeding. I'm going to put it on facultative sexual, but you can have asexual or obligate. Variable is control. That means that the mode of breeding is controlled by the genome itself. Dispersal distance, and then the genome size. It's worth mentioning, I think dispersal distance for archaic reasons is actually inverted. So high dispersal distance means, yes, it says in the reminder there, a high dispersal distance means that the things don't disperse very far. Maybe we should have done something about that at some point, but for now, that as long as people are aware of that. The genome size is the number of 32-bit genome words that exist, and then all of these settings here control what those different words are used for. So, for example, in the default, we have 2 genome words, but only the first one, as in number 0, is used to determine organism fitness. They're both used to determine organism breeding compatibility. So effectively, we have one that is influencing the phenotype with respect to the environment. And then we have the second word is almost a non-coding genome in this setup. We'll leave that as is for now. In the simulation tab, we can change all sorts of things to do with the environment. So what we're going to do is we're going to add those environment files that we just made, which are here. So I just need to select all of these. And RevoSim will load them in a big stack. And you can see that the environment here has changed. We can control the rate at which the environment changes from one file to the other, which is almost like the rate of environmental change. We can control how the environment changes. So if we put it on static, then effectively it doesn't matter that we have 500 images because this one is going to be the only one we see. The environment will not change at all, and organisms will become very well adapted to exactly the color that they find in their environment because that color is permanent and unchanging. More realistically, we might want to put it on one of these other ones. So if we put it on once, it will run through the environment once and then stop. Loops round, bounce, runs through the environment one way and then runs back the other way. We'll put it on bounce for now. Don't tend to touch these things, but you can. The fitness target and settle tolerance terms dictate information about the shape of the fitness landscape. I'm not going to go into the details of that. But in my mind, I've always thought of the lower values of settle tolerance give you a spikier landscape. So you have very large differences in fitness. If the settle tolerance is low, then large differences in fitness will exist between very similar genotypes. Whereas if it's high, you have a much flatter, smoother landscape. The energy input to the system. I'm going to reduce this just to make it run faster. If the energy input is lower, then you have fewer organisms, and so the system is quicker. But yeah, you can modify this here. You can also modify this using the masks feature, which is an additional feature that allows you to overlay an image on top of a particular environmental variable. So if you imagine that being energy, we could overlay an image that is a grayscale gradient from one side to the other, and it would say that effectively the energy at one side of our image is greater than the energy at the other side, and we would be able to see variation in space and time rather than just this fixed global value. But we'll use this for now. And the last tab to show off here is this interactions tab. This contains the pathogen information, which is something I don't play with too much, but which does exist. You can effectively have organisms being killed off by the equivalent of viruses in the background, and then you can have those viruses adapt towards killing off, adapt for virulence if you want them to or not. And if you want to, you can have your organisms stealing energy from one another. So you can turn on interactions to allow them to change the energy of organisms directly by taking energy from one another. Or you can have your interactions modify the fitness, so organisms effectively steal fitness from other organisms and then use that to gather energy from their environment. You can change the number of interactions. You can change the ecological efficiency. So if 100 units of energy were stolen, we could change this to 3. 3 is 30%. And that would mean that if 100 units of energy are stolen, we have 30 units of energy transferred to the predator. But we're going to leave these off for now, just for simplicity. So that's the very quick rundown of all of the features in RevoSim. And I will just, all of the settings, I suppose, in RevoSim. There is more stuff up here, but I will leave that for now. And I'm just going to run this. We've got logging turned on, and we'll see what happens. So there's a vague possibility that everything will die in a bottleneck at the start. It didn't happen. Good. That's nice. It's always awkward when that happens because it's not unrealistic, but it is annoying. And you can see that the organisms rapidly spread out to fill the space, and they rapidly start to adapt to the environment in which they find themselves. So these different colors here, we're in visualization system 1, which means that what we're seeing in this image is just a visualization of the first genome word of every organism, which is the genome word that dictates fitness within its environment. So effectively, this is the coding genome of the organisms. And you can see that you get patches of color, which map to patches of color within the environment. Down here at the bottom, all of our little tooltip summary statistics are ticking over. We have 100,000 organisms, just about, at any given time within our system. Sorry, I'm leaning over again. The mean fitness of those organisms is about 83%. And we've already got 700 species, and in part because that's where we have facultatively sexual breeding, which is going to inflate the number a bit because organisms will tend to form little ephemeral fluffy species. That would be lower if we had obligate sexual. But you can see that these organisms are effectively forming reproductively isolated patches. And by going through some of these systems, I can show you some of the different visualizations you can have. So interactions won't show us anything because we have them turned off. Stolen energy, again, won't show us anything because our interactions are turned off. Pathogens are turned off. Visualization system 2 is the non-coding genome. But if I show you species, these are the species groups. So every color here is arbitrarily assigned to the species when it appears in the simulation. And one color represents a block of one species. And you can see that they map pretty well onto the patches of color within the environment. So we have this purple thing here, which is living in this green blob. This blue thing, which is in the process of going extinct, is living in this gray blob. We have this orange thing here, which is living in a kind of lime green blob over here in the corner. And we're already at what's that? 6,500 iterations just now in the space of since I pressed play. So it's pretty fast, really. And it actually gets faster as the number of species increases because one of the major time-consuming elements is the algorithm that searches through species to detect new species. And that's much faster if there's many small species than if there's one big one. There's a few other settings up here, like we can look at mean fitness. And unsurprisingly, we see that the mean fitness tends to be highest towards the center of blobs because these species are effectively adapted to these blobs. And then the fitness is lower on the boundaries because they're in these boundary environments to which they're less well adapted. But these environments are not sufficiently permanent or large to accommodate their own uniquely well-adapted species. And here we see you get pockets where you get a new environment emerges, nothing's well adapted to it, and then a new species will enter into that environment and become specialized to it. So that's a brief rundown. I'll just quickly show you what some of the logs for this look like. I'm going to stop this and close it. And I will pull up first just the text log. So this is the kind of thing you'll get out if you run the v2 log like I suggested. We have at the top a header which has all of our settings, which is useful when you forget what the settings are that you used. And then what we have are entries for every species. That little S in the box on the left there denotes that this line is telling us information about a species, this little S here. And we have the iteration number at which this occurred. So this is the log for the 900th iteration or 899th iteration. And then this information refers to the population as a whole. So this is saying we have 147 species at this iteration, split between 102,000 individuals, with a mean fitness of about 12. And then this is all of the different species and information about. You can write your own little scripts to extract this and turn it into something a bit more plottable or a bit more user-friendly, but all of the information you want effectively it goes in here. The only other thing is I showed you I said I was turning on the species tracking. I'll show you this here. So this is what you get if you turn on one of those optional visual loggings. Every logging iteration, it will record an image of the panel that we're interested in. So I told it to record an image of the species. So if I turn this on, if I open this up, you can see it's actually unhelpfully small because it's only a 100 by 100 pixel image. There we go. You can see every species, and I can just click through these, and you can see a playback effectively of every species that arose over the course of the simulation and what happened to it. And my clicking through it is faster than my computer is willing to cope with, but there you go. So that's just a rundown of RevoSim and how it works and how easy it is to use. I hope, if nothing else, I've gotten that point across. And if anyone has any more specific questions or indeed more general questions about how this works or what it might be used for, I'd be very happy to take them. But I hope that some of you might be able to go away and consider at least how you might use this in your own work, or even in something like student teaching as well. If you have student projects that you need to run, this is a very easy and user-friendly tool. So I hope that that's helpful, and thank you all for listening.
That was great, Euan. Thanks so much. That's really great. Yeah, so if anybody has any questions, feel free to put them in the chat.